We've spoken before about CRISPR, this amazing tool that can rewrite life's code. Now, imagine using this power against a formidable foe like HIV. HIV is a relentless virus, hiding deep within our cells. Current drugs can control it but cannot remove it completely. This is where CRISPR changes the game. CRISPR is like a precise molecular surgeon, able to find and alter specific DNA sequences. We're moving beyond managing HIV, we're aiming for its complete eradication. The dream of an HIV-free future now beckons, inspiring researchers worldwide. CRISPR technology shines a bright guiding light, illuminating a path we once only imagined. The potential to lift this heavy burden is immense. This is the incredible promise CRISPR holds for us all. So how does CRISPR actually fight the HIV virus? The science behind it is elegant, yet powerful. HIV cleverly inserts its genetic code into ours, becoming an unwelcome part of our DNA. CRISPR offers a sophisticated way to find this hidden enemy. It uses a guide molecule to locate the virus like a highly skilled detective for our genes. Once CRISPR finds the viral DNA, the Cas9 protein gets to work. Cas9 acts like a pair of tiny molecular scissors, making a precise cut in the DNA. This cut can disable the HIV virus or remove it entirely. The CRISPR system is expertly guided by RNA, matching the HIV DNA sequence. It leads the Cas9 protein to the exact spot, like a unique genetic address system. The system then snips out the viral genes, leaving the host cell's DNA to be repaired. Successfully removing the viral genome means the virus can no longer replicate. This is the transformative potential that we see. CRISPR can achieve more than just removing HIV. It can also edit our own genes to make our cells resistant to HIV. One key genetic target is a gene called CCR5. HIV often uses the CCR5 protein as a doorway to enter our immune cells. Blocking this entry is a strategic aim. CRISPR can edit the CCR5 gene, preventing the protein or altering its shape so HIV cannot use it. Some people naturally have a CCR5 mutation, making them highly resistant to HIV infection. CRISPR aims to mimic this natural protection. HIV can lie dormant in cellular reservoirs, remaining inactive for long periods. Current antiretroviral drugs cannot reach this dormant virus. CRISPR might offer assistance by waking up the virus, a strategy called shock and kill. Once the dormant virus becomes active, antiretroviral drugs can attack it. This dual strategy offers a comprehensive attack plan. From the lab to living models promising strides, these innovative ideas sound exciting, but do they work in practice? Scientists have rigorously tested CRISPR against HIV in laboratory dishes. They took human cells infected with HIV and applied CRISPR gene editing technology. The initial results have been very encouraging. They show viral DNA can be precisely cut out. In many lab experiments, CRISPR worked, accurately cutting the viral genome. This action stopped the virus from replicating and, in some instances, completely eliminated HIV. These early successes built confidence for more advanced research. The next step involved animal models, crucial for testing new therapies. Scientists used special mice with human immune cells called humanized mice models. These mice were infected with HIV, and researchers used CRISPR-based approaches to remove HIV. The results from these animal studies are promising. CRISPR treatment successfully reduced viral loads and in some cases eliminated HIV. These studies are paving the way towards human clinical trials for HIV. This is a critical bridge for translation. The horizon of hope challenges and future triumphs. The journey towards an HIV cure is not yet over. There are still significant challenges to address. Delivering CRISPR to all infected cells is hard. We need to ensure it reaches every hiding place. The virus can lurk in many different tissues. We must also avoid off-target effects, ensuring patient safety. Efficiency needs to be further improved. Scientists worldwide are working hard on these issues. New and improved delivery methods are being developed. CRISPR systems are becoming more precise. The future promise of CRISPR remains incredibly bright. We are shifting our focus